What is the largest single cell? What is the longest single cell? What is the longest cell or largest cell? What is your estimation? One single cell ね。longest cell とか largest cell とか what it is?1 minute 1 minute left 1 minute remaining Please finish your work and give your answers to the assistant, okay? Yes. セルだ、セル。Today's topic is about cell. だけど、セルね。And biomechanics about cell. And birth and death of cell. これはadditional part ね。Okay. Yeah, firstly, you have to define what is a cell. I mean, this two parts. 部分おしまい、部分フィニッシュ。What is cell? Cell ってvery difficult to explain, but to some minimum unit for about the living creatures, of course, minimum unit. To live the biologically actively, one living organism, the structure and functional minimum unit. Structural and functional minimum unit. And maybe some cell is separated from inside, outside, and inside is separated by this membrane, some film-like membrane. Yeah? But it is easy to go through this membrane. Some material is also go through via the membrane. Cell body inside of this is called cell body, and some structures, some kinds of various kind of structure is there inside the cell, and the inside of this is not so water-like fluid, not like water-like fluid, but more gel-like. これ、inside of this is なんて書いてあるの？これか。main of this is nucleus. Some nucleus is there. Nucleus is there. So it contains some DNA. It is for to transfer to the next generation. And other organism is there, not only nucleus, but also some Golgi body, lysosome, mitochondria, and other things is there inside of this membrane. But just go back to this part. Yes, single cell. Yeah, maybe the chicken's egg yolk is a single cell. Before the fertilization, only egg yolk is a single cell. Is it true or false? Egg yolk is a single cell or not? Egg っていうの egg 卵 egg なんかこう egg is here. Egg yolk is a single cell. Or multiple cell, single cell, which? Okay? Before fertilization, so therefore it is single cell. Okay? So therefore the cock is this size. But to the, for example, ダチョウって英語でなんていうの？ちょっとこう鳥いるでしょ。not flying bird. In Australia, for example, their egg size is looks like this. Okay? So therefore, this egg yolk is the largest one in the world. Singles as a single cell. Okay? それから何これ？フォックス、スポーン、フロックって言ってたら何だろう？フロックのスポーンってなんかこう、これ each egg is here。なんかこう、フォークのスポーン、こういう感じであるでしょ？ inside water。 で、まあ、this egg is after fertilization, it is multiple cells. So therefore, this is also the multiple cells. ね、ブロック、スポール。これまあ、each of them is about to the 1 millimeter to the 3 millimeter size diameters, but all of them is a multiple cell and also they are multiple cells. And the orange とかレモンとか、このオレンジ、これ these single なんだこれなんていうのグレイン How about this grain? だってさこのオレンジとか、グレープフルーツとかレモンとかなんだっけまあみかんがあってでここに房がこうあってこれはなんかこう each grain contains composed of the grains ね composed of grains How about this grain is a single cell? これはシングルセルですかねこれはシングルセルじゃない not single cell This has a multiple cell It contains inside of this it has many cells It looks like the one single cell but it contains many cells それからなんかシングルセルスリビングクリーチャーズはこのパラメシウムとかボルチセラとかあとはフェリオゾアとかこれ all of this is これ例えばこれはアニマルこれプラントこれはアニマルこれ this is all of them are single cells シングルセルクリーチャーズAnd human sperm is a single cell, and nuclear cell, neuron cell. Neurons is also the single cell, as we discussed last week and the next 2 weeks ago. なんだどうやってこうあってなんかこうスターみたいなのがあってそれからエクソンがこう出ててなんかこうペペペとかペペペとか出てるんだけど This is also the single cell ね So thereforeこのaxon part is some of the nerve cell has more than 例えば like the 30 centimeter length of these axons So therefore this kind of cell is maybe the longest one axon is several millimeter to the several 10 centimeters, more than 30 centimeters in some cases. So therefore, this is maybe the longest one. Yes. それからこのコイというの is a single system. はい。ということで、まあ cell is separated by some membrane. Separated by some membrane. Separated by some membrane. Inside and outside is separated. で、なんだMono-cellular organism っていうのは、まあ、シングルセル、living creature. And multi-cellular organism っていうのは、multi-cell organism. That is a plant or animal. The smallest minimum unit is a cell, and the cell makes some tissue and organ, なんかスタマックとかオルガン、まあ、ブレインも some kind of organ だからセルからセルがミニマム and tissue and organ and the individual person とか individual animal the cell, tissue, organ, individual creatures っていうインディビジュアルオーガニズムということで、モノキュラーセル、パラメシウム、like this。これ、this is シングルセルですね。アメーバっていうのもシングルセルですね。シングルセルです。例えば、パラメシウムの動きを見てみましょう。はい。ですね。Or maybe you may watch
the paramecium
when you are the elementary
at
school
lunchtime.
children video.
I'm using a Nikon 40x plant achromat lens on a Nikon fluophot scope with a Nikon fluophot phase condenser. It shows a number of the internal organelles.
Some energy is in part inside of this and consumed.
Vacuoles and things working around inside of it that I've never actually seen before. Oops, watch out. Stand back.
To waste. Yeah. ここに今いいです
There, did you see that food going in there?
Food.
Gullet.
Eat.
And then into a new food vacuole that moved away. You see that? That's how it eats.
はい、これシングルセルですね。はい。アメーバ見ますか？アメーバ。これなんかゲルでこれ shape is not the fixed in it deform various ways ね。changing the shapes ですね。fluid inside of thisそれから、モノキュラーセルガーオーガニズムの中でユーグリナっていうのがありますね。日本語でベンボンっていうフラチュラっていう this またストリング using this string it moves forward and backward or side movement making. And this part is very important. This is a protein motor is here. So therefore this can move. This can rotate. By making this rotation, this shape is looks like a coil-like shape. And then if it is rotate, it can move like the screwing movement. ですね。これもなんかあるかな。これ見てみますか。これですね。これはシングルセルですね。シングルセルで、このモーター、プロテインモーターです。And also this small hair is around here, and therefore this これはゾウリムシ。この in this case, the small hair is here. Paramecium has a small hair. By waving these small hairs, it can also move like this. はい。 for all of these is the ATP is the energy source. So it looks like the human muscles, animal muscles. ということで、then the next is a multi-cellular organism. And multi-cellular organism is divided, categorized into the plant and animals. Plant and the animals. What is the difference of the plant and animals? ここに書いてあるけど、plant cell has a cell wall. This wall, plant cell wall, is a very rigid, hard. But the animal cells is a very flexible membrane, only film like the membrane. And also the plant cell contains a chloroplast. Chloroplast is some green one. Green one is here, so therefore it looks like a green. So these two is the main difference of this. The plant cell looks like this, and the animal cell looks like this. Okay? ですね。はい。Next はこれですね。じゃあちょっとHear this rap music.
Today's the day we're gonna learn about the cell. If I teach it okay, you'll know it very well. So listen up, sixth graders, no room like our haters. Let's talk about the building blocks of life, cells that make us. Cells, they're made of organelles. Try to pull a fast one, the cytoplasm gels. The nucleus takes over, controlling everything. The party don't stop till the membrane box is seen. Inside the vacuole, we can float around for hours. Running around with chloroplasts, loving sunlight showers. Cells, they're made of organelles. First things first, there's two different types. Animal and plant cells that make up all life. The little things that make up microscopic cells, the main structures there, we call them organelles. Now let's break it down and get some information. How do cells work? It's a crazy combination. Cells, they're made of organelles. Try to pull a fast one, the cytoplasm gels. The nucleus takes over, controlling everything. The party don't stop till the membrane box is seen. Inside the vacuole, we can float around for hours. Running around with chloroplasts, loving sunlight showers. Cells, they're made of organelles.
The cell membrane is the board of control. Who can cross over? The membrane lets you know. The gooey stuff inside is called the cytoplasm. It holds the organelles. Don't worry, cause it has in the middle of the cell. You found the big brain. The nucleus surrounded by nuclear membrane. Don't forget the vacuole is filled up with water. It's busy keeping life.
The bracket's getting hot. Cells, they're made of organelles. Try to pull a fast one, the cytoplasm gels. The nucleus takes over, controlling everything. The party don't stop till the membrane box is seen. Inside the vacuole, we can float around for hours. Running around with chloroplasts, loving sunlight showers. Cells, they're made of organelles.
The mitochondria is something every cell needs, breaking down the food and releasing energy. There's a place inside the cell where chemicals are stored. The squiggly wiggly bodies releasing even more.
Located in the cell is endoplasmic reticulum, synthesizing enzymes for respiration. And none of you still think that this rap is whack? Remember ribosomes making proteins inject. Cells, they're made of organelles. Try to pull a fast one, the cytoplasm gels. The nucleus takes over, controlling everything. The party don't stop till the membrane box is seen. Inside the vacuole, we can float around for hours. Running around with chloroplasts, loving sunlight showers. Cells, they're made of organelles. Looking at the plant cell, weird and green. 2 more cracks is all it takes, you see. Cell wall knows what's up when it comes to keeping structure. Strong cellulose, the cell can give a shocker. CHLO, class. These little green machines are having a blast. Photosynthesize and fill with chlorophyll. They do the cell work, running up a high bill. Cells, they're made of organelles. Try to pull a fast one, the cytoplasm gels. The nucleus takes over, controlling everything. The party don't stop till the membrane box is seen. Inside the vacuole, we can float around for hours. Running around with chloroplasts, loving sunlight showers. Cells, they're made of organelles.
Hi, cell made of organs. ということで、one important thing is plasma membrane. Plasma membrane is it looks like the liquid bilayer. One layer is this is friendly to the water. This side is friendly to the water, but this side is not good. So therefore, the outside of this membrane is the hydrophobic group, and inside of this is a hydrophobic group. And so therefore, the two-layer system is a membrane. Two-layer system is a membrane. But around this membrane, there contains some. The protein is here, and the protein is this part is make some holes. So therefore, some material is goes through this hole, and the sodium pump. This is the sodium pump. So therefore, sometimes it is open, sometimes it is closed. So therefore, the material going through and going outside is make some information. Information transfer is made by this material transformation. Material transfer. And inside the cell is gel-like, the Zoll forms. So therefore, cytoplasm, it says. The Japanese agar or the Chinese isinglass. It looks like the melted down like the liquid form, thick liquid form. There are many protein is inside there, and the gel-like Zoll form. Okay? And one important organism is this mitochondria. In this picture, mitochondria is here. So because it makes some energy from the oxygen, so this mitochondria is a very important part because it makes some energy from the oxygen. So by using this adenosine triphosphate, so therefore it generates the carbon dioxide. So therefore it is said that inside of this cell, it is making the breathing, the cellular respiration. So and then it contains the different DNA compared to the nucleus. So therefore in some assumption or some person like Lynn Marguerite said that it is another living creature is living together inside of this cell, she said. それはどこに書いてあるかというと How mitochondrium create energy と見ています。
Our body is made up of trillions of cells. They all require energy to function. This energy is created within our cells, in the mitochondria. Here, food is converted into chemical energy called ATP. ATP is released by the mitochondria, so cells can use it. Mitochondria consist of two membranes, an outer membrane, separating it from the cytosol, and an inner membrane, surrounding the so-called matrix. The area between these membranes is called the intermembrane space. ATP is generated at the inner membrane of mitochondria by an efficient mechanism called oxidative phosphorylation, involving several membrane protein complexes. Nutrients provide high-energy electrons in the form of NADH, which are used by the protein complexes to pump protons from the matrix to the intermembrane space. This continuous pumping creates a proton gradient, where the positively charged protons are attracted to the more negative matrix. When the protons re-enter the matrix through the ATP synthase protein complex, they catalyze the production of ATP.
it is hard to understand. So therefore このプラズマメンブレンの話をしましょう。このセルトランスポートっていうのを見てみましょう。It's here.
Cell transport is the process of how things move in or out of the cell through the cell membrane. There are two broad categories of cell transport. The first category is passive transport. For a cell, passive transport means it's an automatic process that doesn't require any input of energy. For example, diffusion is a passive process in which particles move either into or out of the cell from an area of higher concentration to an area of lower concentration. The cell doesn't use any energy when this happens.
これは It's common でね、higher density part というから lower density part in some material is moved through if it is some holes or some it is easy to transfer. これはまあ passive way.
The second category of cell transport is active transport. This is when particles move from an area of lower concentration to an area of higher concentration. When particles move against the concentration gradient, energy is required, often to allow protein pumps to assist in particle movement. Why would the cell need to move particles from a low to high concentration and expend energy to do it? An important example is seen in your heart muscle cells. In order for your heart to beat, there are certain molecules that have to move from an area of low concentration to an area of high concentration for those cardiac muscle cells to work. So the main things to remember are passive transport happens automatically with no energy required, while active transport needs energy for it to occur.
だけですね。はいです。これがプラズマメンブレンのワーキングパートね。Through this membrane, the inside and outside cell, some material is transported. And then, for example, in a human cell, there are about 200 kind of cells is there, and the 6 trillion number of cells is there inside of the one single human being. So therefore, the many kinds of cells is there inside the blood, the red blood cell, white blood cell, platelet, and the vessel wall, the vessel endothelial cell, or the smooth muscle cell inside of the vessel wall, and also the muscle cell is also there. It is a skeletal muscle cell and the cardiac muscle cell, and so on. And the skin also several kinds of cell is there, and the bone systems, there are many kinds of cell is there. So various kinds of cell is here inside of the human being. Specialized cellular tissues, just for the mechanism. How cell becomes a specialized.
We've mentioned a lot about specialized cells. Specialized plant cells, specialized animal cells, so many kinds of specialized cells, it's going to get a bit crowded here. But have you ever wondered, how do they get specialized? How does a neuron or a muscle cell in your body have the structure and function they have? I mean, can you imagine if they had to switch jobs for a day? That wouldn't go so well. They're so specialized for the function they perform. Well, this video is going to talk about how cells differentiate into other cells, which basically means how cells become specialized. Remember that many multicellular organisms, like a plant or you, well, these organisms come from a fertilized egg cell. Let's take a look at a human fertilized egg, otherwise known as a zygote. Well, that zygote divides to make more cells. And more cells. Oh look, it's a morula. And more cells. Oh look, it's a blastocyst now. You know, the problem is, if the cells just keep dividing, if you remember from our mitosis video, that makes identical cells. Well, that's great for growth, and so dividing is definitely going to happen, but that alone is not going to result in different specialized cells with different specialized functions. There's something else that will be happening for that. So let's take a look at that in a bit of detail. We're going to pause here, the blastocyst. Notice it has stem cells, and these stem cells, they're amazing. See, they're not differentiated yet. They're not specialized. They're like blank slates. They don't have a special structure. They don't have a special job. They can become any type of body cell. Now, a reminder about body cells in your body. They all, with a few exceptions, contain all of your DNA. So neurons and muscle cells in your body, they don't have different DNA. They use different parts of your DNA. Genes are regulated, which means the genes can be turned on and off. It's important to understand because that's a big part of how these stem cells are going to specialize. Stem cells will activate certain genes in the process of differentiating into different types of cells. Transcription factors are major key players here. They're typically, but not always, proteins, and they determine which areas of the DNA code will get transcribed into mRNA. Now that mRNA can eventually be used to make specific proteins that can impact what a cell is going to look like and what a cell is going to do. That means transcription factors have a major role in determining which genes are expressed in a cell. Because a cell that's going to become a skin cell is going to have different areas of genes expressed than a cell that's going to become a stomach cell. There are both internal and external cues for stem cells, which can involve these transcription factors. Examples? Okay, an example of an internal cue could be transcription factors present in the cytoplasm of the original starting zygote cell, which will eventually be present in the cells that come from it. Now, the specific location of the stem cell within the developing embryo can matter because the transcription factors available in different areas of the developing embryo can differ in quantity and type, which could impact what a stem cell differentiates into. External cues could involve cell signaling from other cells next to it. External cues can even be environmental effects like temperature. There's still a lot of research in this area, and we can't wait to see what scientists discover about this in the next decade. So, stem cells are the unspecialized, undifferentiated cells that can become other cells in your body. But not all stem cells are found in a developing embryo. Stem cells can also be found in your body as well, like your muscles, skin, liver, bone marrow, just to name a few. These are often called somatic stem cells. To give some relevance to this, it's likely you've heard of bone marrow transplants before. Well,
いやそんなもんですね。Stem cell is one original one. It has some many potentials to become some specialized cells. And the gene, and which part of the gene is activated is important to separate it into the specialized cells. And then what is the cue to become specialized is one important research area, and not yet clearly discovered how to manipulate some specific cell into. So therefore, まあもちろん inside factor, internal factor and external factor is there. Internal factor is some conditions, and the external factor is some stimuli, like temperature or pressure or some kind of stimuli is important. So therefore, how to differentiate this kind of cells is one important, not yet discovered area. If we can do that, we can create some specific organs from the stem cells, but not yet realized. Okay? From this part, the manipulation of the cell. The manipulation of the cell is a mechanical manipulation, like the aspiration of the contact or insertion or the drilling or cutting or removal some part, and the DNA injected inside of this, or implant some organ inside one cell, like this. So the mechanical manipulation is one way to operate the cell manipulation. The second one is electrical operations. Electrical operations, some potential is applied, some current is applied, some electric stimuli, and also heating by the electrical way is also there. And the optical manipulation is one important one is the optical trap, the laser tweezers. Laser is applied and some cell is attached to this light, and the light, when we light moved, the cell is moved followed by this light movement. So therefore, yes, optical tweezers, you should watch this. Alright, some light is applied, and then this cell is deformed. And also some light is moved. This cell is also moved following the light movement. これだけですね。ディフォーメーションってやってますね。ちょっとこれ、インターションオプティカルトラッキングっていうちょっと見てみましょう。これはサミー・ユニバーシティ・ラボラトリーズ。
Welcome to the Bioinstrumentation Teaching Lab at the MIT Department of Biological Engineering. I'm Steve Wasserman.
And I'm Stephen Nagel.
I assume you've tuned into this video because you like to make quantitative measurements with laser tweezers. If you were looking for cute kitten videos, you've made a horrible mistake.
Optical tweezers can apply measurable forces to micron-scale dielectric particles. This capability offers a unique and valuable tool to manipulate cells and cell components, as well as measure mechanical properties at the micron scale. For example, instruments just like this one have been used extensively to investigate the mechanical properties of biological polymers, as well as the force generation mechanisms of molecular motors. In many cases, the force is actually applied to a microsphere which is attached to the molecule of interest, as a way of providing a convenient handle.
To make quantitative measurements, the instrument records the displacement of a trapped particle over time. The exerted force is very nearly proportional to the displacement, so the trap can be modeled as a linear spring. Accurate force and position measurements depend on careful calibration of the position detector responsivity R and the trap stiffness alpha, also called the spring constant.
The properties of the trap are a function of the laser power, the bead diameter, bead composition, as well as the optical properties of the surrounding medium. In this video, we will show you 3 different methods to calibrate the trap.
Let's start with a review of the single beam gradient force laser trap. In this system, a high numerical aperture lens brings the laser beam to a sharp focus. Light rays entering a microsphere at the center of the trap are refracted. An example ray is shown at the extreme right of the beam. Refraction changes the momentum of the photons in the beam, as indicated by the green arrow. The particle undergoes an equal but opposite change in momentum. The ray refracts again as it exits the particle, which adds to the force on the microsphere. Some of the photons reflect off of the microsphere. These generate a momentum change in the opposite direction. The overall force on the particle can be approximated by adding up the contributions of many rays. For a particle in the center of the trap, all of the forces sum to 0. If the microsphere moves out of the center of the trap, the refracted light also changes direction. The forces are no longer in balance. There's a net force on the particle toward the middle of the trap, so the force exerted by the light tends to restore a particle to the center of the trap. The strength of this kind of optical trap depends greatly on the gradient of the light intensity, so it is important to use a well-aligned instrument to achieve best results. The optical trap used in this demonstration was developed here in the instrumentation lab. If you are considering making a trap of your own, Thor Labs offers a kit that includes all of the bits needed. Search for part number OTKB on the Thor Labs website. The position detector is sold separately as part number OTKB-FM. This is a block diagram of the instrument. The trap uses a 100x 1.25 Na oil immersion microscope objective for both trapping and imaging. It's designated L1 in the diagram. The condenser lens, designated L2, is a 10x 0.25 Na microscope objective, 2x, which is in the infrared range, out of the trap component of the trap, and then subtracting the stock trap. Calibration involves simply mere frequencies. A trap F naught, P naught, over which to sample the displacement. The force on the particle is equal to 3 pi eta dV as before, and this must balance the trap force.
というわけです。まあいいや。スキップ。もちろんchemical manipulation is also there. But you may be mostly familiar with this kind of mechanical tools. Yes, needle is inserted inside of this cell. For example, yes, you can see this. Yes, this cell is by using this micropipette inserted in here, like this, and then some gene or some nucleus is from here, removed here and implanted inside of here. Another way is this kind of moving operations, and then this is a piezoelectric motor is used, and so therefore it more smoothly the inside of this cell through the membrane. This smooth insertion of the micropipette is the one of this the University of Tokyo's laboratory more than 20 years ago. By using this, but in many cases using this kind of gear system is made, and this is a stick to move this insertion needles, and then the other fluid method is also there. In this case, the joystick operation is here and connected to this fluid tube, and then this part is moved like this. Micro-manipulation. Left side, right side, micro-manipulation system. ですね。これでインジェクションします。これはオプティカルシステムですね。これはピペット is used to capture the cell, one cell, single cell ですね。 まあいいや、まあ、こういうふうなシステムがあるということですね。これが終わって、それから next is なんだっけ、バイオモレキュラーか、メカニカルプロパティのメジャーメントメソッドで、it is for example one method is using the ATM atomic force microscope でcantilever is used, so therefore the deformation of cantilever and the force applied to of this cantilever is measured, and then the mechanical features is force and stretch curve is measured. And also the optical tweezers is used because trapping power is related to the power of the light. So therefore by using the tweezing force and the displacement is measured, and then the deformation feature of the cell membrane is measured, for example. And also the glass needle is used, and also gel solutions property is measured by using the sound propagation speed and so on. I will explain the next more in detail. Like this. これも同じものなので、はい。で、dynamic property is because the one cell is usually floated inside some liquid, but if it is only single cell is given on the plate, this deforming. So therefore the time and deformation, so therefore the surface force is measured, estimated by using this curve. Then firstly, this looks like this, but this has the gel is filled, and then the gravity also applied to this because some weight is here. So therefore the membrane feature and the inside of liquid feature is estimated by using this curve. Okay? Another method is actively the two plate is pressed from the both sides, and then this force and this displacement is measured. So therefore the inside pressure of this the plasma membrane is estimated by using applied force and deformation height. And then the feature of inside of this plasma membrane is estimated. Another method is the one single hole is here, some liquid is filled both two sides, and this cell is deforming. If it is changing the level difference of slight difference of both sides, height difference gives some pressure difference of this side. So therefore in this case, this side, right side is more higher pressure, so therefore it is pushed here, and this gel is moved little by little. So this array shape is deformed depending on this the difference of the pressure of the liquid. So therefore this time deforming is measured, is observed, and then the characteristic of this plasma membrane is estimated. This is called capillary method. And in this case, some particle is inserted inside of this plasma membrane. And in this case, this is a small iron is inserted here, and some magnetic force, magnetic field is applied. The magnetic field is applied. The 0 to some magnetic field is applied, and then released. So in this case, this iron material is moved by this magnetic field because some force is given here in this period, and so therefore the movement of this is a little bit, but when this force is released, become zero, but this is a little bit moved and the state like here. Because not only the spring constant, but also some viscosity is also there inside of this gel. So therefore, the viscoelasticity property is estimated by using this curve. Okay? Yes. Done. それからなんかピペット。ピペット is here. This is a one cell, and then the pipette is give some negative pressure here. So therefore, the deforming. This part is deformed, slightly deformed. The pressure is given here, and this membrane part is moved like this, and then if the pressure is released, it can go back to the original position. So this deformation, the force and the deformation is measured, and then this is also the viscosity, viscoelasticity is estimated of this plasma membrane, and also the cytoplasm. Yes. This is micro-pipette technique. It is called micro-pipette technique. So and this is a study example over here. In this micro-pipette method, this is a result of this method. The negative pressure is given here, and this deformation part. When the difference of the pressure is given, and the deformation length is here. So this is a one cell result, and whatever this is. This is a vascular cell because the blood is flowing here, and this vascular cell is here. The inside of this vascular tube, this cell's mechanical feature is depending on this blood flow pressure force. When the pressure force is always higher, this cell is become more harder. And this pressure force is become lower. This cell's mechanical feature is become more soft. Okay? So therefore, when the high shearing stress is always given here, this cell become more harder, and low shearing force condition, this cell becomes more soft. So therefore, it is measured the mechanical feature of this one single cell. これ見てみますか？これはこのピペットをここのところにつけて、これはdeveloping some the test bed of this, the measuring of this single cells the feature. It's explaining. それは何ですかと言ったら、これ近づけていって、this is approaching to this cell and the suction like this. And they are making some the negative pressure, and then the normal part, usual part, and the deformation is measured. これが、so by using this image processing, this part of the deformation is measured by this like this. というのこのディフォーメーションをオートマティカリーメジャーっていうシステムをディベロップメントしましたと言って This is deformation data is this deforming like this, okay? So therefore the pressure is given here and given here, next pressure, and so this deforming like this. The end of this part is shown like this, okay? っていうのがワンメジャーリングメソッドですね。はい、これです。っていう感じでした。で、by using this, this kind of the curve is the measured. And the other example is here. This is これは押し込み棒って書いてあるから3セルになんかこのsome material is pushed into and the deformation is observed. Some material is pushed to this cell and then the deformation is measured ということでこれFirst trial, second trial, some data is here. The length of pushed in and the pressure force is applied force is given here. And then this curve is measured. これはどんなやつですかって言ったら、これですね。これは ATM で感知レバーでこのそこにプッシングする。これピペットで holding this cell.And then this is pushed inside in here, and this deformation is measured. And then this cantilever, so therefore some applied force is measured in this level. ですね。はい。これが一つの例で、違う例もDon't remember the difference. これは another test case. でこれピペットでホールディングしてでプッシングです。これみたいな結果ですか Similar result. はい、というやつがあって、あとはthird example is this is pipette, and one single cell is here. Inside of this liquid, the cell is living, and this pipette is, in this case, pipette is the holding here, holding pipette, and the next pipette is holding here, and holding, and then this pipette is moved. Okay? So therefore the applied force and the deformation of だからデルタ X is the measure here. So this is like some only like the tension test. One holding and the one actuator is moved like this. So therefore the displacement and the force is measured by this the piezoelectric force measurement system is here, and the movement is here. So therefore the length stretching and the applied force is measured. The case 1, 2, 3, 4, 5, 6, it looks like the similar curve is observed. ですね。はい。です。So, therefore, by using several methods, the mechanical features is measured. For example, red blood cells case, the G is the elasticity coefficient, spring coefficient, and the mu is elasticity coefficient. It looks like this. And this is a white blood cell, and the fibroblast cell looks like this. And another method, elastic modules and viscosity coefficient is measured like this. And then in other tests, the fibroblast cell and the nerve cell, some nerve cell and T3 cell is measured. And then this viscosity is measured. Then one single cell's mechanical feature is measured. And then the final part is only the additional part, fertilization. I have shown one of this video before this class. And then the division, regeneration of the cell, because this is a birth of cell, and this is after living, after created. How to generate the next one. This is one method is cell division, and the other method is cell regeneration. Divided inside, yeah. And then I have said from the stem cell, it has some potential, and it becomes a specialized cell by the cell division and cell reproduction. And so therefore the meiosis and mitosis is you have learned in maybe the high school period, maybe. Animal cell is making look like this, and the plant cell is the making looks like this. As in plant cell's case and animal cell's case is looks like this. And then maybe the telomere is you may be interested in, because the cell division, the copying some DNA, but the end part has some telomere part is there. Telomere part is not completely copied and become shorter, and then this telomere part is become shorter and shorter, and the it is telomere part is become shorter, and then this is a little bit shorter, not copied again. So therefore this telomere part is set the coupon ticket of the livings. So the number of cell division is limited by the length of this telomere part. But the Escherichia coli is used for the DNA operations, because this cell has no telomere part. So therefore it can be copied again and again. So therefore it is used for some the DNA operations. And then the final part is the death of the cell. How the cell is disappeared. One is apoptosis, the plant death, and one is necrosis. It is some damaged and become sick, and then this cell is destroyed. Necrosis, apoptosis はまあ見ますか。はい。これさっき見た。ね。まあバクテリアとかでデストロイされてしまうんですね。 は、まあ、メンブレンがブローク。アポトーシスはplant is because some part of the cell is disappeared because of some operations. In some cases, it is separated into multiple cells, but so the necrosis is, in many cases, is the cell is broken. Some kind of injured, or some radiated, or some dystrophies injured, or the frozen injured, or bed sores. It also one of the necrosis part. But apoptosis is also important because, for example, this is a cock's hand and duck's hand. The cock's hand, yes, it is similar to the human fingers. Firstly, it has some. This is the webbing part is webbing part is remaining like this duck. But after becoming some growing, this part of the cell is dead. So therefore, you can move the finger individually. Yes, this is similar to the human being. Human being is also inside the mother's body. This part is also there. But the file become growing. This part is disappeared because of this part of the cell is become dead. Okay? So therefore, some plant apoptosis is happened, and then this part is individually movement is realized. Okay, but this mechanism is not discovered well, not understood well. Then why this apoptosis is happened, and then they become separated. The movement is also achieved is not understood well. Yes. So that is all. Maybe that is the end of what I have prepared. Okay. はい。ということで、もう一つまた again announcement is next week there have been some exhibition about welfare technologies, welfare equipment. So therefore, if you are interested in, especially the last week's content and the 2 weeks ago's content, it is related so much. So therefore, if you are interested in, you should attend. The Saturday is also opened in the near the Kokura station. And then the IPS International Symposium, the International Collaboration Symposium will be held at the end of this month. So therefore some keynote speeches there. This is held on the Thursday and Friday. So if you have some time, you should attend to this keynote speech or the invited talks and the poster sessions of your senior student. Okay.